Okay, so a second class of large biological molecules or macromolecules are lipids. So lipids are large biological macromolecules, but they are not true polymers. So they are the one kind that I said they're important, but they're not necessarily polymers. So they're not like very cleanly made of like monomer, 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 monomer. They are actually very diverse molecules. And the one thing that unites them all is that they mix poorly, if at all, with water. So that's kind of like the unifying thing of lipids, not that they have the exact same building blocks. The unifying feature of lipids is that they mix poorly, if at all, with water. And that is because they consist mostly of hydrocarbon regions. So if you remember, water is a polar molecule. It has parts of it that are partially positively charged, parts that are partially negatively charged. So water molecules are polar. And so things that are polar are going to be like attracted to water molecules, and they'll bind and dissolve really well. And so those kinds of things are hydrophilic. They like water. But things that are nonpolar and have really long hydrocarbon regions, those are nonpolar, and so they're hydrophobic. They do not mix well with water. So yeah, that's part of what make something hydrophobic, because they have regions that are nonpolar, like hydrocarbons. And so you, might not have heard about lipids that much, but there are some kinds of lipids that you might have heard about. So the most biologically important lipids are fats, phospholipids, and steroids. And so I'm going to break down each one of these, or give examples of each one. So fats, you've probably heard of. And then again, that's another thing that you might have seen as it relates to like our diet. In nutrition facts labels, you can see the fats and fat content. And so as a molecule, fats are made from two types of smaller molecules. A molecule called glycerol, and then a molecule called a fatty acid. So glycerol has a specific structure. It looks like this in the gray right here. And so glycerol stuck to a fatty acid makes a fat. So a fatty acid has one structure, but it has to be stuck to a glycerol to be considered a fat. And there are different kinds of fatty acids. In this case, it's called palmitic acid. So glycerol plus a fatty acid makes a fat. Glycerol, is a 3-carbon alcohol with a hydroxyl group attached to each carbon, so an OH on each carbon. And then a fatty acid has a long carbon skeleton. It could vary how long it is. And then a carboxyl group at the end. So a carbon double bonded to an oxygen. So again, remember, some of these are going to come up over and over. Fat is made of those two parts. And fats separate from water because water molecules hydrogen bond to each other. Again, they're polar, and so they hydrogen bond to each other, and they exclude the fats because fats are nonpolar. So in a fat, there's usually 3 fatty acids joined to that glycerol by what's called an ester linkage. And so if there are 3 fatty acids joined together, this makes a triglyceride or triacyl glycerol. And so that might sound familiar if you've gotten, a blood panel done recently, in a lipid panel. Triglycerides are something that are measured in things like that, in a big picture sense again. So triglycerides, there's a glycerol and then 3 fatty acids. And the fatty acids in a fat can all be the same fatty acid, or they could be 2 different fatty acids, or even 3 different fatty acids. So here in this picture on the top, shows, you know, a fat coming together, a glycerol molecule, and then a dehydration reaction adding on this fatty acid. And so that's how you add a fatty acid onto the glycerol, is by this dehydration reaction. So again, even though this isn't technically a true polymer, that's still how you would add things on. And then in this bottom panel, panel B, this is a fat molecule, a triacylglycerol with 3 fatty acids. And in this case, 2 of these fatty acids are the same fatty acid, and then this one is different. And then this is what's called an ester linkage. So the fatty acids can vary in the length, you know, like how many carbons there are in a row, and in the number of double bonds, if there are any, and where those double bonds are. And so you might have heard of like saturated fats versus unsaturated fats. So part of that is because of the fatty acids. Fatty acids can be saturated, and so they'll have the maximum number of hydrogen atoms possible and no double bonds. So let me move forward for a second. So a saturated fatty acid, like highlighted it in yellow, there are no double bonds, and so that means each carbon is going to be, you know, connected to another carbon, and then there will be 2 hydrogens, and so they're connected to the maximum number of hydrogens possible in this molecule. And so in this space filling model, the little black circles represent the carbons, the little white circles represent the hydrogens. So there's the maximum possible number of hydrogens that are here on this fatty acid, and so it is saturated with hydrogens, like it's full with as many hydrogens as possible. And then an unsaturated fat has a double bond somewhere. So that means that those 2 electrons, 2 pairs of electrons are getting shared, and so there are not some hydrogens there taking up those spots. And so it's unsaturated with hydrogens, like it doesn't have as many possible hydrogens as possible. So it's unsaturated, and then because of the double bond, it does kind of make a little break in the chain, like a bend. Yeah, so unsaturated fatty acids can have 1 or more double bonds, whereas saturated fats have no double bonds, and there's the maximum number of hydrogens possible. So those are the fatty acids. So fats made from the saturated fatty acids are called saturated fats, and they tend to be solid at room temperature. Most animal fats are saturated. So if we come back to this picture, so if this is a fat, because it has all saturated fatty acids, they line up really cleanly. And so because all of those fatty acids line up really like linear and they stack really well together, that's kind of what makes saturated fats able to be solid at room temperature, because they're like so stacked and pressed together. But unsaturated fatty acids have bends in them, and then so unsaturated fats are gonna like not sit together so packed, and so they're gonna be more liquid, these molecules, and so unsaturated fats tend to be liquid at room temperature. So fats made from unsaturated fatty acids are called unsaturated fats, or oils as well, and they are liquid at room temperature. And typically plant fats and fish fats are usually unsaturated. And so yeah, you might have heard of these again when it comes to like the big picture and diet and nutrition. So saturated fats and unsaturated fats. A diet rich in saturated fats, you know, especially because they're so like solid and they pack together, can contribute to cardiovascular disease through plaque deposits. And so in some products, there is a process called hydrogenation where you're adding hydrogens. So say something like an unsaturated fat that's kind of like an oily, but for some reason this company wants it to be able to be like solid at room temperature. So hydrogenation is how you can like add hydrogens to unsaturated fats. So now they have more hydrogens and they have, you know, now they're saturated. But doing this can also create what are called trans fats. And so trans fats are, is there a picture? No. So trans fats are still bad for you. So by doing this process of adding hydrogens, it also creates trans fats. And this may also contribute to cardiovascular disease, and actually more than having saturated fats. So yeah, these trans fats may contribute more than saturated fats to cardiovascular disease. So that is, you know, like the big picture that you might have heard of some kinds of lipids. And so in general, the major function of fats is energy storage. So in our body, when we have like excess amounts of energy, then our bodies store it in the form of fats in our adipose cells. So humans and other mammals store their long-term food reserves in adipose cells. In adipose tissue, you know, we store the energy there. But adipose tissue also cushions vital organs and insulates the body. So yeah, when you're kind of tying these concepts together, carbohydrates are important because they do provide power for your cells and energy. But carbohydrates kind of get a bad rap because if we eat too many carbohydrates or anything really, like if we're taking in too much energy in general than we're using, then our bodies have to store it. And so typically our bodies store it in the form of fats. So yeah, it's that we're taking in kind of too much energy than we're using. And then so that's kind of why carbs get a bad rap. But it's also that we're not eating like a balanced meal and we're not using all the energy that we are typically taking in. So yeah, fats, the major function is energy storage long term. Another kind of lipid are phospholipids. So in a phospholipid, there are two fatty acids connected to a glycerol that also has a phosphate group. So it's two fatty acids and a phosphate group that are attached to that glycerol. And so those two fatty acid tails, again, they're basically hydrocarbons. They're hydrophobic because they're hydrocarbons and non-polar. But that phosphate group at the head of it, attached to the glycerol, is polar. And so that makes it hydrophilic. It likes mixing with water. So phospholipids are interesting that they have parts that are both hydrophobic and hydrophilic. So this is how we typically show, you know, just little cartoons of phospholipids. It's like the hydrophilic head and then two little tails. But this is how it like actually looks, the structural formula and, you know, the space filling model. So there's the two fatty acid tails that are just long chains of hydrocarbons, hydrophobic. But this glycerol and the phosphate group, and then actually there's this extra part. All this is polar, and so it is hydrophilic. It likes mixing with water. So phospholipids are kind of unique in that they have parts that are hydrophobic and hydrophilic. And so they actually make a pretty big part of our cells, the membrane of cells. So there's what's called a phospholipid bilayer. There's 2 layers of phospholipids. And yeah, it's really interesting that this happens. If you just dump a bunch of phospholipids in water, they self-assemble into this double layer sheet. So because it's like a double layer, it's called a phospholipid bilayer, bi meaning 2. And so at the surface of a cell, you know, phospholipids are also arranged in a bilayer. And again, cells on the inside and outside of cells, it's a watery environment. And so it kind of makes sense that all the hydrophilic heads will be sticking out and facing the watery environment of either inside the cell or outside the cell. And then the hydrophobic tails that don't like water, they kind of just like face each other and hang out with each other. Sometimes I kind of compare it to like extroverts and introverts. So, you know, extroverts really get along with people and they're going to be like the people-facing kind of people. And so the hydrophilic heads are kind of like extroverts. They like mixing with water. And then the hydrophobic tails are kind of like introverts that don't really like mixing with other people. But if you were to shove a bunch of introverts in a room, they'd probably end up hanging out together to not really talk to each other. So yeah, all the hydrophobic tails will kind of just orient and face each other so that they're not really talking to the water or mixing with the water. So it's really nifty that this happens because this provides like a separation from the inside of the cell and the outside of the cell. So this phospholipid bilayer helps form the boundary between the cell and its external environment. It makes sense, you know, if there's a watery environment on the outside and a watery environment inside, how do you make a separation is with like a setup like this. So that's phospholipids. And then the third kind of major important lipid are steroids. So steroids have a carbon skeleton that have 4 fused rings. And so notice they're not all the same shape. So these are like hexagons and then there's a pentagon. So steroids have a carbon skeleton. These are all carbons at the end, remember? And there's 4 fused rings. And so one important kind of steroid is cholesterol. So cholesterol is a type of steroid and it's a component in actually the cell membrane also. So when in a future lecture we talk in more detail about the cell membrane, you'll see how cholesterol is kind of part of that, even though it's not a phospholipid. Cholesterol is an important part in cell membranes in animals. And it's actually also a precursor from which other steroids are made. So you start out with the cholesterol and then you can just kind of address that and then make other steroids. And it can also make some hormones. So you might have also heard, of course, of cholesterol in the big picture sense. A high level of cholesterol in the blood may contribute to cardiovascular disease. So in one way it's important for your cells, but you also don't want too much of it because it can also lead to cardiovascular disease. So this is a summary of lipids. There are triacyl glycerols, or fats or oils. It has the glycerol and 3 fatty acids, and they're an important energy source. There are phospholipids, and they're important in the cell membrane, the lipid bilayer. And then there are steroids, and they are also important parts of cell membranes, but they can also make signaling molecules that travel through the body, in other words, hormones. So that's a summary of lipids.